speak a little bit about my work about chromosome instability in cancer. So I hope to place this topic a little bit into a historical perspective. And so I thought I'd show you these images. This is David von Hansemann, who was a German clinician scientist around the 19th century. And so he was the first to draw really these images where he noticed that the cancer cells might missegregate chromosomes while they divide. And so we call this chromosome instability, and so it's basically the frequent missegregation of chromosomes during cell division or mitosis, and it results in aneuploidy. And so aneuploidy is really the state of cells that have abnormal chromosome numbers, whereas chromosome instability is like the dynamic process of the missegregation of chromosomes. So this is a fairly basic picture of different stages of the cell cycle, and as the cells progress through the cell cycle, really where the chromosome segregation happens is during mitosis. And so that's really where the chromosome instability happens. Also, I should probably say that the type of chromosome instability that we're interested in is really whole chromosome instability, so that's the missegregation of whole chromosomes. And then there's also a different kind of chromosome instability, which is called structural chromosome instability, and that involves breakages of chromosomes and translocations and those type of abnormalities, but that's not what we are focusing on. So that's chromosome segregation happening during mitosis. So when a cell enters mitosis, here at the first stage of mitosis, prophase and prometaphase, at that stage the chromosomes have been duplicated, and so there are two copies of each chromosome, and these two copies are held together by these ring structures indicated in purple here that are called cohesins. And now, as long as the kinetochores here at the centrosomes of the chromosomes are not occupied by these microtubules that emanate from opposite poles of the cell, a so-called wave anaphase signal is generated at those unoccupied kinetochores, and it prevents the cells from entering anaphase prematurely. So at metaphase then, when all those kinetochores are occupied by microtubules, this wave anaphase signal is extinguished, and so that enables then the cell to ensue anaphase, and that means that a sequence of reactions is started, which results in the cleavage of these ring structure of these cohesins, and then the two copies of each chromosome are allowed to move to opposite poles of the cell. And then following cytokinesis, which is the physical separation of cells, which is not really shown here, that will be the stage after this, you will then end up with two cells that each have two mDNA content. Now, as I said, chromosome instability is the missegregation of chromosomes, so that means that that you will end up with aneuploid cells, so some cells having gained an extra copy and other cells having lost a copy of a chromosome. So this checkpoint that I spoke about is called the mitotic checkpoint because it really makes sure that that chromosomes do not prematurely segregate. And when we talk about the mechanisms of chromosome instability, over the past several decades, I would say, a number of different mechanisms have been identified, one of which is actually defects in this mitotic checkpoint. So chromosome instability is promoted by defects in mitotic checkpoint signaling, but also by centrosome duplication defects. Cancer cells often have over-duplicated centrosomes, so they have more than the normal number of two centrosomes. So the centrosomes here are located here and here. These are the microtubule organizing centers during mitosis. And so you can imagine that if you have more than two copies, that then these microtubules will attach to different kinetochores of chromosomes, and it becomes a big mass, and that also leads to chromosome instability. Similarly, sister chromatin cohesion is sometimes defective in cancer cells. And so I mentioned these cohesin ring structures. Sometimes defects, not necessarily in these cohesins themselves, but usually of some upstream regulators. When they are defective, it leads to premature segregation of chromosomes. So you can imagine that if this cell were now to proceed to anaphase, then you also will end up with a situation like this, and so with aneuploid cells. And then there's cytokinesis. I briefly mentioned that cytokinesis is the physical separation of the cells at the very end of mitosis. And so you can imagine that if these cells and eventually not physically separating, then you will have cells that become binuclear, that really have 4M instead of 2M DNA content, and that has been shown to also facilitate as an intermediate process of really more missegregation and aneuploidy events. So this kind of summarizes that a little bit. This is again the cell cycle where most of the defects that lead to chromosome instability occur in mitosis. So there's this cohesion defects, mitotic checkpoint defects, cytokinesis failure. Centrosome duplication defects really happen actually in S phase, because during S phase centrosomes are duplicated. So those defects happen really in S phase, where you get over-duplicated centrosomes. But then the missegregation events will occur in mitosis. And another defect is DNA replication stress that has recently been found to lead to chromosome instability. So one term that has been introduced a few years ago to explain how cancer cells become unstable or how they become aneuploid is called oncogene-induced mitotic stress. And so the basic principle of this is that common defects in cancer, such as p53 mutations or amplification of MYC or loss of RB, signal through a few common signaling intermediate regulators, as it's called here, that leads to defects in transcription and phosphorylation events of a whole host of different proteins. And so, since a lot of these proteins are involved in regulating mitotic checkpoint signaling, centrosome duplication, and histochromatic cohesion, you can imagine that that some of those common mutations in cancers lead to this, as it's called, mitotic stress that then contributes to chromosome instability. So I guess the summary version of this is that that common defects in cancer cells lead to chromosome-induced mitotic stress, and that results in chromosome instability. And one thing that I wanted to use as an example today is some of our studies that I think is really a good example of this, which involves defective signaling of the RB pathway through a protein called EMI1, and that via this route promotes chromosome instability. So EMI1 stands for early mitotic inhibitor 1, and so this is a cell cycle regulator. It's also an E2F target gene, so you can imagine that if the RB or retinoblastoma pathway is defective, then it leads to upregulation of E2F target genes, including EMI1. And so consistent with that, we see that if we look at a large variety of different cancers, we see that EMI1 is often overexpressed. This is just an example in glioblastoma, but we have looked at a whole host of other cancers, comprising a total of 7,400 tumor samples and nearly 1,500 next normal control samples. And so we see that in these 115 independent studies, 89% of the cases EMI1 is overexpressed. And what's quite interesting is that there's a clear difference between solid and hematological or blood cancers, because we see that this percentage is really 95% in solid cancers, whereas for blood cancers it's about 50-50, and this is a very highly statistically significant difference. So this shows that EMI1 is overexpressed in most human solid cancers. We have also looked at the role of EMI1 overexpression, how that relates to patient survival. And we see in the survival curve here that high levels of EMI1 constitute a poor prognostic factor in breast cancer. But this is not limited to breast cancer. We also see it in ovarian cancer and in lung cancer, as well as in a few other cancers. And so this indicates that EMI1 overexpression is really a marker for patient prognosis in multiple cancers. So all this, of course, is just correlative and not really positive. So we really wanted to see whether EMI1 overexpression might actually, in an in vivo situation, promote chromosome instability. And so what we did is we generated a transgenic mouse model to overexpress EMI1 in vivo. And so what we saw was a number of different things. We saw that EMI1 overexpression in these mice causes a variety of different cancers. And some examples include splenic sarcoma, lymph node sarcoma, thymic lymphoma, hepatocellular carcinoma, and lung adenoma. And then we went on to look at also how the survival of these mice was affected. And we see that EMI1 overexpressing mice develop tumors at relatively long latencies, but they do so earlier than wild-type control or isogenic control mice. And I say isogenic because we overexpressed EMI1 here in an inducible way, so we could do an isogenic control. And then we also see that the EMI1 overexpressing mice compared to the controls, that more of those animals develop tumors, so there's an increased penetrance. We see that those tumors are on average larger. We see that there's an increased tumor burden, so in other words, the EMI1 overexpressing mice develop more tumors per mouse as compared to the control mice. We see that there's an increase in different forms of cancer because we compared lymphomas, sarcomas, carcinomas, and adenomas separately in these mouse strains. And what we see is that in the EMI1 overexpressing mice, these frequencies are elevated in all cases, except only for the carcinomas and adenomas. These differences were statistically significant. Consistent with that, we also see that if we look at the carcinoma-specific tumor burden, so the number of carcinomas per mouse is significantly increased in EMI1 overexpressing mice compared to controls, and that's also true for the adenomas. And it's not statistically significant for the lymphomas or as I show here, the lymphomas and the sarcomas. So that's really reflected on two levels here, both in frequency as well as in terms of tumor burden. Another thing that we observed is that there is a significant increase in liver cancer specifically in these mice. We see that there's a significant increase in metastatic load. So of the mice that do develop cancer, a significantly larger proportion proceeds to develop metastasis. And we see that there's an increased hyperplasia or neoplasia that we see in the tissues of these mice. And so these could be precursors of cancer. And then finally, consistent with also what we see in patients, there's a significant increase in the number of immunological abnormalities and the frequency of those in the EMI1 overexpressing mice. And so this pertains to inflammation, for example, and also infiltration of lymphocytes into tissues. So this really shows that the in vivo overexpression of EMI1 promotes tumorigenesis. Now, I mentioned that we are interested in chromosome instability specifically, and so we had some indications that led us to think that perhaps EMI1 overexpression could promote chromosome instability. So we would really wanted to look at the mechanism. And so what we did is we first looked at the tumors of these mice, and we used FISH for this, so fluorescence in situ hybridization. And what we found is that the EMI1 overexpressing mice, that the tumors are highly aneuploid, whereas in control mice that is not the case. And if you quantify this, you see that that is highly statistically significant. So this indicates that EMI1 overexpressing tumors are highly aneuploid. Now, we then wanted to look more at the cellular level at what kind of chromosomal abnormalities or abnormalities pertaining to segregation of chromosomes we might find. And so what we found is when we looked at these tumors in these mice, that there were cells that progressed through anaphase normally. So you see here the different chromosomal masses segregating to opposite poles of the cell. So the subset was normal, but then we also found that there were cells that had lagging chromosomes. Here's an example, which is an indication for chromosomal segregation. We saw that there are chromosomes that were misaligned or outside of the main masses. For example, here you see chromosomal material. Then we had cells that we called monopolar, and we looked at this also using immunofluorescence. And so what you can see here is that the chromosomes are condensed, but they are kind of in like an astral shape, monopolar spindle-like. And we also found a fraction of cells that had tripolar, so it suggested that there were 3 poles in these cells. So that could be indicative of overduplication of the centrosomes. So when we quantified this, we saw that in the control tumors there was a subset of cells that showed the chromosomal abnormalities, the ones that I pointed out here above. But in the overexpressing tumors, this was significantly increased, so increasing from about 20% to about 80%. And we also saw that with the exception of the tripolar phenotype, that the increase in each of these different phenotypes was statistically significant. And so that really indicated that EMR1 overexpression promotes lagging chromosomes, misaligned chromosomes, and also the formation of monopolar spindles. And so that was all the mouse work. We then also went back to human cancers again, because we really wondered if there might be a correlation or an association between the degree of chromosome instability in human cancers and the level of EMR1 expression. And so what we did is we used what is called the SIN70 score. This stands for the chromosome instability signature comprising 70 different genes, which has been shown to accurately reflect the degree of aneuploidy in human cancers. And so we basically just plotted that against the expression level of EMI1. And in this case, in breast cancer, we saw that there was a very significant correlation between the degree of EMI1 expression and the degree of chromosome instability in these breast cancers. And so we then were curious to know whether this also was the case for other cancers. And so in addition to breast cancer, we looked at other cancers and we saw that that was actually the case. And so we noted that this was true also for ovarian cancer, for glioblastoma, low-grade glioma, colon cancer, kidney cancer, lung cancer, and endometrial cancer. And so this really showed that degree of EMI1 expression tightly correlates with the degree of aneuploidy in human tumors. And we actually went one step beyond that and found that EMI1 actually ranks in the 75th percentile of the 70 genes that are used for this signature. So really indicating that EMI1 itself is a much better indicator of chromosome instability than most of these 70 genes. So just to summarize this part, so we saw that EMI1 overexpression occurs at high frequency in human solid tumors, and that it strongly correlates with poor patient prognosis, that it promotes tumorigenesis in vivo, so really indicating that it is causative rather than just correlative, and we see that it promotes chromosome instability in vivo situation in the mice, and that it is a powerful marker for chromosome instability in human cancers. So now coming back to the more general picture again, I showed that pointer's not really working anymore, but I showed that if you look all the way to the right, that EMI1 overexpression is really an example of the more general oncogene-induced mitotic stress model that has been proposed. Also, I wanted to draw your attention to the fact that chromosome instability can also be tumor suppressive, because as much as chromosome instability can lead to the gain of oncogenes and the loss of tumor suppressor genes, the reverse can also happen. And so there are also tumor suppressive effects of chromosome instability, which could be cell autonomous and non-cell autonomous at the same time. And so there's a whole number of different factors that could play a role. At the bottom here, you see the 8 hallmarks of cancer as they have been defined. And so there's a lot of interaction between chromosome instability and how that can lead to various abnormalities and how it can be tumor suppressive as well as oncogenic. So I think from a clinical perspective, I think that the following is actually important to realize as well. So this kind of is a follow-up on this oncogene mitotic stress. So if you look at chromosome segregation in general, the gene expression or the activity of those genes or proteins, that can vary from really 0 to low, normal, high, and extreme. All those are observed in cancer. Actually the middle, I should say, is probably not observed, because usually the complete loss of these genes is cell lethal. But what we do see is that if you look at the degree of chromosome instability, that's usually when the levels are lower of these genes, then you would see a low or a high degree of chromosome instability. In the normal situation, everything is normal, of course, and then if there's a high expression of a lot of, or activity of a lot of these chromosome segregation genes, you see also a lower high degree of chromosome instability. But it has been observed that if there is a really very high or extreme degree of chromosome instability or gene activity, that it leads to extreme chromosome instability, and that actually is tumor suppressive. And so that's what I hope to show here. So if you look really at the patient level, you see that that the complete loss of these genes usually does not occur. But when you have lower or higher degrees of chromosome instability, that usually confers a poor prognosis for patients. And when chromosome instability is normal, that usually is a good prognosis. But it has also been shown that that if you have very high levels of chromosome instability, that it actually confers a good prognosis because usually the cells are not able to handle extreme chromosome instability and they would simply die. So that's really the final slide that I hoped to show to you, except for, of course, the announcements. I'd like to thank all the people involved in our group, as well as a few collaborators. And then I listed my email address here in case you'd like to contact me about any of this work. And then there's also, of course, the funding agencies, the NHMRC in Australia, the National Breast Cancer Foundation, and some institutional funding that really is supporting our work. So with that, I'd like to thank you, and I'd be happy to take any questions.
So I thought you were showing that there is also structural aneuploidy with the, if you're seeing all these different forms during the mitosis, doesn't that mean it's not only whole chromosomes, but also chromosome arms and chromosome regions that you're actually breaking?
Yeah, that's a good question actually. To some extent they're also inextricably linked because it has been shown that when chromosomes missegregate during mitosis, there are sometimes breaks occurring. And so it can lead to loss of parts of chromosomes, which could lead to micronuclei formation, for example, and also to translocations. And so to some extent it's really inextricably linked. Our main focus of research is on the whole chromosome missegregation, but I also showed in one of the images, I don't know if I can get it back here, that here that it can also lead to structural chromosome abnormalities, and so yeah, it's kind of all linked. So yeah. Other questions?
I think we're running out of time.
Okay.
It was a wonderful thing.
Thank you.
But we must live along.
Absolutely. Thanks.
To